Randy Hyer today from Moderna, Senior Vice President for Vaccine from Moderna. Very big event for us, very big day. Very happy he's going to come. So I've already mentioned Dr. Hyer's background. He received his medical training at Duke University, got his PhD from the University of Oxford, and his MPH from Harvard University. Should I go through with my jokes again too?
Yes, please.
But now, Gene, would you mind bringing me over the bag and the book? Because unfortunately, we can't give Dr. Hyer the kind of honorarium that we'd like to, And usually, we have an extremely generous, over-the-top honorarium, but we can't. So we want to show our gratitude in another way for this talk, which is called "The Beginning and End of COVID-19." Maybe that title is a little premature. The end part of it, I guess we'll hear. I hope not. So first of all, we always give a book to our speakers, and it turns out that almost every book we have he read, but there was one he hadn't read, and that is the book by Barry Blumberg called "The Hepatitis B: The Hunt for a Killer Virus," and this is one of our handful of remaining signed copies. So this is one book he hasn't read. Obviously, he can write his own book about his own virus. That is Randy. We also have, and over the years, I forgot to tell you about Randy, how I got to know him. He led the approval of the Dynavax vaccine. He was head of I guess CMO, or head of the clinical trials Dynavax, which got the first hepatitis B vaccine approved since the originals 30 years ago, and it's the 2-dose vaccine, and it was a big deal. And he led that, and so we may have actually given you this tie, didn't we? Oh, good. All right. So this tie is actually, as you know, the electron micrograph of the hepatitis B virus taken by Manfred Bayer with Barry Blumberg, and it shows the subviral particles. Now it's yours, and there's some other swag in here too. The hat. So without further ado, and we'll add that time to your. Thank you, Randy. I'll turn over to you, and I will lower the lights.
Thank you.
Wait, one more thing. For those of you listening online, we're following your questions, I hope, on the computer, and we'll answer them, But thank you for joining us online. We have several hundred people who logged in online on the two venues. So with that, Randy, thank you.
Wow. Hi, everybody. Well, it's certainly an honor to be here, and thank you, Tim, for that kind introduction. So I'm Randy Hyer. I'm Senior Vice President at Moderna. Talk today about the beginning and the end of COVID-19. I really wish I was talking about the end of COVID-19. I'm sure we all would be in that camp. As you know, we're not there yet. However, we've made some important strides in getting there. And I'm going to talk about the story on how we got to where we are today. And I'll talk a little bit about the future. Where I see it, I don't have a crystal ball. That would be required to know what's going to happen next, I think. However, we got some insights from the virus and insights from other diseases, other pandemics, and so on. But I'm going to start first with more of us. Let's talk about the beginning a little bit. And the first thing I have to show you, Moderna is a public company, so our forward-looking statement, this is, I can make this available to you if you want to read it in detail. This is the story. How did we go from January 11th, 2020, where the sequence was deposited and confirmed on the websites of this virus, new virus, SARS-CoV-2, to an emergency use authorization granted on 12/18/2020? For any vaccine preventable disease, the fact that there's the same year on any slide is a miracle in itself. Okay. And that's really the miracle, and I'm going to call it a miracle that I'm going to talk about today. And why did that happen? How did that happen? How did we get to where we are now? And it was enabled by a number of things. One was the science. People understood the science. The biology of coronavirus was fairly well established. People understood them. The science of inducing mRNA, using mRNA to induce immune response had been established. So actually, the COVID-19 vaccine that Moderna manufactures and markets under emergency use authorization, I believe is the 9th or 10th vaccine for Moderna that we had developed. Now, the other ones weren't so public and they weren't marketed, but they were developed against different diseases. So we had done this for about 10 years. already. And of course, using mRNA as a potential therapeutic had been described first back in the 1990s, just no one had quite figured out how to do it yet. But without question, there was an unmet need and shared sense of urgency. I don't need to explain that any further. And then the collaborations with the partners, BARDA, NIH, PPD, the PPD is a contract manufacturing organization, the clarity from FDA about what was needed, when was needed, the open conversations, and of course the US government stepping in and de-risking the procedure with the advanced purchase agreements, allowing us to make decisions at risk and move forward. This is what got us. And really that's the story, the Epicurve there I now show in the middle. Of course, it started at 0 in the United States. And what's, I think, really interesting is you see how that first, I'll call it the second wave because there was a wave in the middle of 2020. But that big wave there, the October, November, December last year, 2020, where the case counts really started to take off. That's actually when the vaccines, ours and that of Pfizer, went into phase 3 studies. So if you have an event-driven analysis, the one thing that will accelerate the progression of your study is events. And we had events. I mean, it's not what anybody wanted, of course, but it was very useful to get the vaccine, get to our interim analysis endpoint and get the data, and get to the first interim. And then, of course, that led to the emergency use authorization on the 18th. So that is the story. I could almost end here, but I want to tell you the rest of the story. I do need to mention this. The Moderna COVID-19 vaccine is not approved nor licensed by the US FDA. It's authorized for emergency use only. And this is under an emergency use authorization to prevent coronavirus disease 19, known as COVID-19, in individuals 18 and older. There is no FDA approved vaccine right now to prevent COVID-19. This EUA is in effect for the duration of the declaration justifying emergency use. And if you want more information, we have websites available here that have the full prescribing information and fact sheet on the vaccine. So let's talk a little bit. There's the culprit. It's called a coronavirus because if you look at that electron micrograph and you sort of like, just like the sun, you know, the sun has a corona. Well, it's the corona there. These, of course, are the spike proteins. This is the business end of the virus. This is the part of the virus that interacts with the receptor on the surface of many of our cells, known as the angiotensin converting enzyme number 2, or ACE2 receptor. These are envelope positive sense single-stranded RNA viruses. They have a tremendous amount of variability and a high recombination rate, which allows them to easily go back and forth between animal species. And these viruses, the SARS-CoV-2 is very similar to ones that cause SARS and MERS. So I'll tell you a story. I was at the World Health Organization back in 2003 when we had this mystery pneumonia developing in southeastern China. Nobody knew what it was. We were sitting there looking at each other. We had no idea what the agent was. This, of course, became known as SARS eventually, SARS-1. We had no idea what we were looking for. We thought it was some transmissible agent. It had this fairly high case fatality rate. It was actually 10%. SARS-CoV-2 is more like 1% or lower, so it's a very different animal. It also produced symptoms in almost anybody that got infected, so we were able to figure out who had it, and that allowed us to control the epidemic using just public health measures. There was no vaccine. There still isn't a vaccine against SARS-1. And here we are, you know, SARS-CoV-2 came up. Of course, one of the things when they saw those individuals with unusual pneumonias in southern China, what was the first thing everybody thought it could be? SARS. So you use SARS primers looking for it. Well, that's how we got SARS-CoV-2 so quickly. One of the reasons. I mean, not the only reason, but one of them. Of course, so back to coronaviruses, and one animal species can potentially mutate and infect humans and rapidly spread. This is what we saw for MERS. MERS is the Middle Eastern Respiratory Syndrome, very similar. It has a higher case fatality rate, actually, a little more scary, but it tends to be very geographically localized. All of these viruses were thought to have originated in bats and then spread to a secondary species. SARS-CoV-2, we have seen go back and forth, went to minks for a while, came back into people. This may be a mechanism it uses to gain virulence because it can use these other animals as mixing vehicles to gain other genes and so on. And of course, the classic symptoms of the disease we know as COVID-19, coronavirus infectious disease 2019, is basically an upper respiratory syndrome, a cold, fever, cough, shortness of breath, and some muscle aches. Who hasn't experienced that? Then, of course, with SARS-CoV-2 and COVID-19, there can be this sort of lower respiratory infection, and almost infection, lower respiratory disease. it's very immune mediated. And this, of course, leads to the severe COVID-19 that can often result in hospitalization, intensive care unit visits, mechanical ventilation, and even death. The other thing about SARS-CoV-2, which is remarkable, is that the infections are asymptomatic, especially initially. So the virus gets there, it's replicating in a way you don't even know you have it. You're walking around, and of course, the virus is transmitting. This may be one of the mechanisms, the latest variant, Delta, of concern that's literally sweeping the world right now in terms of its prevalence, may be able to replicate faster. So during that initial asymptomatic period, the Delta virus may simply have more variants to spread around the world that may be related to its enhanced spread. Okay. This is an Epicurve of the world. It's actually only out to May of 2021. It's not so much the data, the case counts, but the waves that I'm interested in showing here. So the case counts, of course, started back in January. We had some initial rise there in 2020. Of course, this was quite dramatic because it started from zero. And then we had a very big wave. This was the wave where the vaccines were developed and the case counts were high enough to be able to do the appropriate analyses. Vaccines roll out, and of course we had the cases went down, and then we get another wave, and there's probably another wave going on right now. And so this is kind of classic for respiratory outbreaks, as you get these waves of infection, and we may be experiencing that right now with the Delta variant. It is global in its distribution, but let me give you a really important tip here, is that a case of SARS-CoV-2 or COVID-19 requires somebody who's sick and a positive test. Okay, no positive test, no COVID-19. Well, how much testing do you think is going on perhaps in sub-Saharan Africa or in some of these other remote areas of the world? Not as much. So even though these areas look like they don't have maybe a lot of disease, like, gosh, Africa has no disease, I don't believe that's the case. In fact, if I talk to the experts in there, that in some cases we're looking at 60% seropositivity. So they actually it's already there or already has been there. That may be, I don't know if that's good or bad news. It may be good news in terms of existing herd immunity. It may be very good news that when a vaccine does get there, that there would be an ability to boost on existing immunity. So time will tell. But it's important to recognize that the current global distribution models are based on a test, okay? All right. So let's talk about the vaccine. The technology is very simple. It's the basic of all biology. DNA gets transcribed into mRNA. mRNA is translated into protein. And what we're doing is we're stepping over the DNA step, and we're administering the mRNA to produce a very specific protein. And in this case, a protein that hopefully many of us haven't seen yet. And we're using it to trigger the immune system to develop immune response, so then when we do see it for real, we're ready. That's really what it is. And the way the other thing that's very important is that the mRNA cannot go into the nucleus. These mRNAs do not have the sequences that are required for that. So it cannot integrate itself into DNA and change the cell. Okay? It's a one-way street. And the mRNAs, they self-degrade within a matter of days to hours, and the proteins they make also degrade within a matter of days. So and that's how the body controls a lot of mechanisms, by natural degradation. So our platform to use that has three key steps. We have the mRNA, of course, with the chemistry, the sequence engineering. That's very important to get the sequence correct. The targeting elements to make sure it goes where we want it to go. Then the delivery, the vehicle, the suitcase, if you will, to deliver the mRNA into the cell, which requires lipids. The chemistry of the lipids is very important. The composition, and of course, the surface properties of the lipid nanoparticle. And then we have to marry the two together. We have to marry the messenger RNA and the LNP together to get to the final product, which is what is injected into the arm. So what's in this thing, this lipid nanoparticle, which was really the key as to how this got going. It's a nanoparticle, and guess what? It's measured in nanometers. It's 50 to 142 nanometers, obviously pretty small. It consists of multiple components, including an aminolipid, phospholipid, cholesterol, and a polyethylene glycol lipid. All these are important for the efficient delivery and stability. And what do you look for when you make an LNP? You want to make sure that you get maximum cellular intake. You're trying to get the mRNA into the cell of interest to enable that efficient release of mRNA for a stable drug product and be dosed at therapeutically relevant levels. So it's very simple. It's an injection into the muscle like many vaccines, the LNP goes to the draining lymph nodes, goes through the lymphatic system to the draining lymph nodes, where the antigen presenting cells take it up. They process it like they would many other mRNAs. It's inside a cell. There must be some 200,000 mRNAs in active transcription at any given time, and we're just a very small percentage of those. And then it makes the protein. The protein goes to the cell surface, and you get the immune response. And I'll show you that in a slide in a second. Yeah, there's the mRNA going into the cell. Gone. Okay. So the beauty of that is you can do all kinds of things with it. And we're very busy. So not only can we make a COVID-19 vaccine, which of course is what I'm going to talk about in detail today, we can also make vaccines for other products, influenza, RSV, CMV, EBV, SARS-CoV-2, of course, Zika. Ever remember Zika? We can also make a cancer vaccine, personalized cancer vaccine, using your own tumor antigens to make a vaccine against. Intratumoral immunology, various IO agents that we can actually manufacture, and localized agents, systemic agents. It goes on and on and on. These are all, of course, investigational. The only one that has authorization at the moment is the SARS-CoV-2. But we're busy on all of these at the moment. And I think that this is one of the lasting legacies of the COVID-19 pandemic of 2019 to whenever it ends, is that is the development and the birth and the proliferation of this mRNA technology that's going to change medicine. It already has, but it's going to change it even further in ways we haven't thought of yet. Because with this technology, I can take a protein of interest and put it where I want it, when I want it. And that's a very powerful place to be. So I'm just going to give you an idea. This is just the world record here, really, of from sequence identification to first clinical study. We settled on our final sequence on January 13th. So as you know, the virus had started, there were some unusual pneumonias in December in China. It probably started, I think they think patient zero is in either late November or early December. And these were being reported in Chinese social media, which didn't get a lot of international attention for obvious reasons. And then there was a publication in late December where it was taken from the Chinese and put into the English chat room called ProMed. And that got picked up by the local WHO regional office, sent to headquarters, and that's really how the rest of the world became so aware of it. And then that sequence, the virus, of course, was sequenced. That sequence was deposited on various genome database sequencing systems, including some of those at NIH. And Moderna became aware of it. So after it was finally shared on January 11th, we chose our sequence on January 13th, and then 63 days later, that vaccine was in somebody's arm, which is a world record. Okay. I'm amazed how fast that is. And there's lots of reasons for that. I can go into those. A lot of work had been done in advance. A lot of work had been done behind the scenes. There was a close cooperation with the NIH, the Vaccine Research Center. It goes on and on. But it's just a remarkable achievement to do that alone. Then, here we go. Now we got clinical studies development going on. And of course, from April, we went through a phase 1, an overlapping phase 2, an overlapping phase 3. We were able to do an interim analysis, demonstrate vaccine efficacy, meet the performance requirements, meet the safety requirements, and that led to the emergency use authorization of the COVID-19 vaccine on December 18th from Moderna. We published our results on December 30th, showing an overall vaccine efficacy of an astonishing 94.1% against symptomatic SARS-CoV-2 infection and disease, and 100% against severe COVID-19 disease, which is why we're here today. Let's talk a little bit about it. How does it work? Okay, so there's our friendly lipid nanoparticle. It's got the mRNA in it. It's been injected into your arm, into my arm. It's traveled via the lymphatic systems. It's made its way to the draining lymph nodes, and here it arrives at the antigen-presenting cell. What does it do? Well, it goes in. It's taken up. The mRNA is released. The mRNA goes to the endoplasmic reticulum. It finds a ribosome. The ribosome starts translating into protein. That protein goes to the Golgi apparatus. The viral spike protein is assembled, and it appears on the surface of the antigen-presenting cell. Immune system comes along and sees it and starts an immune response. And this is the basis of virtually any infection. So it's actually mimicking the natural infection process very closely, which is, I think, one of the reasons we're seeing such good immunogenicity and immune performance. I'm going to talk a little bit today. Our studies were supported greatly by our preclinical data. These were in, we saw very good data in non-human primates and mice and other models of SARS-CoV-2 infection and COVID-19 disease. We had very good antibody responses in adult mice and non-human primates, showing a Th1 dominant phenotype. That's important for a safety factor because it's important that you induce a Th1 dominant response versus a Th2 dominant response. And then we saw very robust protection against viral pulmonary challenge in mice and viral pulmonary nasal challenge in non-human primates, again, with no indication of enhanced respiratory disease. That would be the Th1, Th2 story, after viral challenge in non-human primates, even using subprotective doses of the vaccine. So it looked very good at this point. Today, I'm going to talk a lot about binding versus neutralizing antibodies. So what is the difference? Well, first of all, it's an antibody. That's that Y-shaped molecule there. With the business ends on the end here, they sort of, this is the part where the antibody connects with things, including the SARS-CoV-2 virus. Interesting, antibody is, I think, about 1,200 amino acids, and the spike protein is very similar in terms of size. In fact, the size of the spike protein in amino acid residues on the SARS-CoV-2 virus is 1,273. That happens to be the name of our investigational vaccine, mRNA 1273. So that's how it was named. So you heard it here first. At any rate, so the binding antibody is an antibody that binds to foreign protein. And the way I was trying to explain this to somebody one day, an analogy came really clear to me, is that binding and neutralizing antibodies. Because neutralizing antibodies, of course, prevent the virus from causing infection. They're neutralizing, okay, and they're important. Binding just stick to the virus because the body doesn't know which protein matters. And it's the equivalent of taking an apple and dipping it into hot chocolate sauce, okay? And then pulling up, you see all the chocolate dripping off it? Well, those are all binding antibodies, ones that stick to the apple. Those are all binding. They're binding to something. They don't know what. And then some of those will prevent the virus from doing its job. Those are the neutralizing antibodies. That's sort of the difference. So we looked at these both, at the binding and the neutralizing, as important indicators of the body's response to the foreign protein. So this is the development program. Of course, remember back in 2020, we got it into humans, the phase 1, we call it P101 study. It was our first in human dose ranging study to make sure we got the dose right. And then what's interesting here is that the phase 2 was overlapping. Normally in development, this phase 2 wouldn't start till out here somewhere. In fact, it wouldn't even have started yet in normal development. And that's because you have to de-risk this, because all this requires tremendous expense. You know, clinical study is a very, very big expenditure, particularly for a small biotech. So the fact that the US government stepped in and said that we're going to guarantee a purchase, assuming it meets certain performance criteria, allowed us to overlap these studies. And then after we saw what we needed to see in the phase 1, we were able to commence the phase 2, and very similarly for the phase 3. And that, of course, that phase 3 led to an emergency use authorization in December of 2020 at an interim analysis. And of course, we're continuing that study in a follow-up period because of the importance of safety. And then we have other studies going on. We're studying in adolescents. And that study actually has an interim analysis been reported. It's under consideration right now for emergency use. We have a study ongoing in younger people and children, and we also have some variant studies in progress I'll talk about in a minute. But the reason that what it's important here is to note is there were no shortcuts taken, okay? The vaccine went through this, had to meet the same safety and efficacy standards with the FDA they would have done for any other vaccine. The reason that we were able to do it quickly is that by it being de-risked and also the close cooperation of everybody allowed us to overlap the studies to accelerate it along. And this was the secret, is that one of the reasons we were able to do it so quickly. Okay. In the phase 1, first thing you measure is you're going to have a new vaccine against a new disease. Well, what do you measure it against? You know, how do you know your in a development study? How do you know what dose to use? What are you going to measure against? And what you use, you look at antibody titers in people who've had the disease. The idea is that, well, they've recovered from the disease, so whatever they have now must have worked, right? So we want to meet or exceed those antibody titers. And that's a lot of how the vaccines were developed, against convalescent sera. Now, it's not a perfect model. It happens to be the best model we had. So we started with our phase 1 looking at the antibody titers to people against convalescent disease. Now here we show the, here's the titers. we also looked at, I'll just go here. We also looked at T cell response because, again, I mentioned to you how important the phenotypic response of the T cell is important, that it's a Th1 versus a Th2 response. And these data here, though quite complicated, do show that in 3 different age groups, that the predominant T cell response shown here was a Th1 CD4 T cell response, which is what you wanted to see by looking at the various cytokines that are produced in the T cell populations. And that was important from a safety viewpoint because prior vaccines in some time ago now had, in some cases, had developed a predominantly Th2 response, which resulted in vaccine-enhanced disease, which of course we didn't want to do. But what's important is that this was a Th1 response, and we've seen no vaccine-enhanced disease in either our animal studies or in people at all to date. So we're pretty confident we're on the right side of this page. We did look, of course, at antibody responses. This is what I want to show you here. These are neutralization antibody responses. And this is kind of interesting because this is now looking into the future, really, from that time point, is on the left is our neutralizing assay, and on the right is our IgG or our binding antibody assays. What's important is that you can see after the doses shown here with the little arrows that the antibody response was first of all robust. This is logarithmic scale. And importantly, it stays high over a period of time. This is actually out to 6 months for both the neutralizing antibodies and the binding antibodies, suggesting that the immune response is robust and persists. And of course, that's very important given what we're dealing with. So our key takeaways is from our phase 1 study, we saw neutralizing titers in 100% of evaluated participants 14 days after the second dose. That was obviously, yes, reassuring. It was clear that the 100 microgram dose, this was a dose ranging study, was the dose of greatest interest. It did have that Th1 response, of course, and we had that persistence of antibodies, which was very important. So this now we can overlap into the phase 2 study. Now this is the protocol here. This study, of course, is obviously fully enrolled, has been analyzed. It was really a dose study between 50 and 100 micrograms to make sure we got the right dose going forward. These were all placebo-controlled studies, of course. And this is important here. This shows the binding antibodies and the neutralizing antibodies at the various time points after assessed. And what's important here, what you to take away is you see that far right column, that's sort of the dark color. That's the convalescent serum antibodies for either binding on the left or neutralizing on the right. And as you can see that initially the titers are lower than the either the neutralizing or the binding titers, but with the subsequent dosing and time period, in all cases, the titers exceeded that of the convalescent sera. And that's what we wanted to see, of course, to go forward. We do monitor safety very closely, especially when we were trying to identify which vaccine and the dose to use. This shows the various symptoms. I think all of us have probably experienced some degree of adverse reaction, that is the pain or the fatigue or the reactions you feel. In all cases, and actually in all of our studies to date, those have always been short duration, usually mild or moderate, very few above that level, resolve in a couple of days, and showing that even though the vaccine is reactogenic, it still is well tolerated and was able to go forward with it. And I'll show safety data throughout, but that was a very important point about looking at the, and this has allowed us to monitor the difference between the 100 micrograms and the 50 microgram dosing. What we saw was that the 100 and 50 were actually pretty well tolerated, suggesting we could use the 100. And in the 100, we saw a more persistent, more consistent production of neutralizing and binding antibody titers above those of the convalescent sera. So the 100 microgram dose was chosen for the phase 3. And that's what that tells us. And that's essentially what I summarized here from this slide. So let me go on to the phase 3 because that's the important one. Now the phase 3, this is the pivotal study. This is where all the chips are on the table. This is where you play for keeps and so on to find out, is your vaccine going to meet success criteria to where it might be available for either authorization or ultimately approval. It was a very simple study. We took 30,000 people, randomized them 1-to-1. They got either our vaccine, mRNA 1273, remember 1,273 amino acids in the spike protein, or they got saline, 2 injections. And we didn't know, of course, who got what. And that's the important part. It was done across the country at many different sites. We tried to make it geographically diverse and also go to areas where we knew COVID-19 was active. More importantly, in fact, really importantly, was that the trial had a strong component of diversity and inclusion, because we knew that COVID-19 was disproportionate in who was affecting the most, either by occupation or by ethnic and racial background. So we were very aggressive in our diversity inclusion to make sure that the trial represented the United States as best we could and the people in the United States who are getting this awful disease, so that our data would be a very good indicator of what the vaccine could do in the populations most affected. And so you can see here, if you can see the faces, of course, but you can see the breakdown. In our 30,000 study, we had 6,000 with Hispanic ethnicity, 25% over 65, number of educated students, roughly equal male, female, for 3,000 African Americans, over 8,000 people living with chronic disease, 22% healthcare workers, almost 2,000 retail, restaurant, and hospitality workers. So we tried to make sure that the trial represented the public as best we could. Looking at safety, it's very important. This is the solicited local ads. We're asking, did you hurt, did it hurt, and so on, within 7 days after each dose in all participants. And as you can see, the primary local adverse reaction, of course, was pain, as I think we have probably all experienced. I know I have. And it went up a little bit with dosing. This is dose 1, dose 2. The gray is the placebo arm. But overall, again, most were grade 1 or grade 2, very few grade 3 reactions, otherwise, very well tolerated. Looking at systemic reactions, this, of course, is the, fatigue, headache, myalgia, arthralgia effect that I think many of us have probably experienced. I know I have. This was a little bit more pronounced when you went from dose 1 to dose 2, but again, there were very few grade 3 reactions, and most of these self-resolved within 2 to 3 days. In our study, in our clinical study, 96% of the people who got the first dose went on to get the second dose, suggesting that reactogenicity, though unpleasant, was not a factor in getting both doses. This is looking at adverse events. So this is events that occur after that or during the reactogenicity or after that reactogenicity. And what's important here is that the ratios, the percentages in the placebo group and in the mRNA 1273, the investigational vaccine group, were very similar across overall reporting of adverse events. And the placebo group was 21.6, we were 23.9. And if you run your eyes down the table, you can see that the percentages were very similar between the two groups. Okay, moving from safety, now we can move into efficacy. And this, of course, is the number of the shot heard around the world, if you will. When we did the case splits, when we unblinded the study, it was in late November, the first interim analysis, we saw an astonishing 94.1% vaccine efficacy with very tight confidence intervals, showing 11 cases that had occurred in the vaccine group and 185 had occurred in the placebo group. So pretty strong evidence the vaccine was doing something. And then when we went into people with severe COVID-19 with a definition of severe COVID-19, we saw no cases in the vaccine group and 30 in the placebo group, giving a point efficacy of 100%. So very, very encouraging data, obviously, which was the basis of the emergency use authorization. This is the Kaplan-Meier curve over time. So the question is, when did the cases start accruing? We started counting, of course, at the second injection. And we started counting that's 14 days after the second injection to give the vaccine ample time to work. And as you can see, shortly thereafter, the cases in the placebo group started to climb. The people in the vaccine group was much lower and stayed much lower over time. And then very importantly, when we did an analysis across subgroups, not just subgroups of age, because we know that age is a risk factor for COVID-19 and severe COVID-19, these are the point efficacy estimates shown here with the little dots, even the 100% being the far right. You can see that the efficacy remained very high. In fact, if we looked at, and even with people with comorbidities, it still remained very high, which was encouraging. No difference between men and women. Small difference between people who are considered to be at risk for severe COVID-19, which we might anticipate. But what I thought was really attractive was when we looked at racial and ethnic group breakdown, comparing just people of white race versus communities of color, you can see that the point efficacy estimate remained very high in communities of color. In fact, it was actually a little bit higher at the point estimate. So this was very encouraging that the vaccine was working across a number of populations and where it needed to be. Wow. That's where we got to, okay? Then what happened? Well, the virus mutated. Virus said, I'm not done. And mutations occur, okay? They're a natural byproduct They can emerge, and a mutation or a variant can emerge. It can disappear. Maybe it's not viable, and we don't even know about it. We have seen multiple variants throughout the evolution of this pandemic. I suspect there will be more to come. Many of these mutations do not affect the virus's ability to spread or cause disease because they don't alter the interaction between the virus and the host being us. And those mutations are eventually competed away, and the other ones survive. And interestingly, way back in early 2020, a variant of SARS-CoV-2 with a mutation at position number 614 with a D to G switch in the amino acid residue started, and that variant is still in virtually every viral protein sequence to date. The vaccine was generated against a virus sequence that did not have this mutation, but it doesn't have any effect on the efficacy of the vaccine, obviously. If we want to drill in on the variants a little bit, there's been a few of them out there. B117, the names keep changing. I usually have trouble keeping up with them. This is known as the alpha variant now, using the Greek system. And this is the amino acid residue, or this is a spike protein, and it's a trimer. So there's actually 3 identical parts. That's the color. So this, of course, is a 2-dimensional representation of a 3-dimensional object. But the blue, the purple, and the brown, it's really a trimer. And this shows where the mutations have occurred that gave rise to this alpha variant, the dominance of the alpha variant, which at one point was the dominant variant was clearly pushing off all the other variants off the map in infection and prevalence infection studies done. And the receptor binding domain is the very tip up here. This is the business end of the virus, where it actually interacts with the ACE2 receptor. As you can see, there was a mutation right there in the RBD, or receptor binding domain. This other thing called the N-terminal domain, or NTD, also had a number of mutations. These mutations, we don't know the true function of the N-terminal domain. We think it facilitates the entry of the virus into the cell. The RBD sticks to the cell, and the NTD somehow helps it get in. And then what I think is really interesting is if you overlay the human ACE2 receptor shown here in this yellow, this ribbon, protein ribbon, you can see the 501Y mutation is right there in that pocket, and it's interacting with the receptor. So it probably alters somehow the attachment of the virus to the cell. We know that the 117 was more transmissible. Just in the interest of time, these are additional pictures of the spike protein. I think the one that's most interesting is this one here that I have circling with the mouse. This is looking right down on top of it. So you can see it's a trimer with the 3 identical parts, and the receptor binding domain being right here in the middle. And then, of course, the B117 or alpha variant came along, and as you can see here in this graph, particularly in the United Kingdom, within just a matter of a few weeks, it pushed all the other variants right off the map. And this, of course, is what we're seeing with the latest variant with Delta, and I suspect will continue as variants emerge and gain their fitness. The virus gains fitness. Then along came a variant in South Africa, originally identified in South Africa, known as the B1351 or beta variant. And this one was kind of scary because, first of all, look, it's got 3 mutations in the receptor binding domain now. And if we zero in a little bit, you can see that one of those mutations, the so-called E484K, which many people call the EEK mutation, is interacting directly with, look how close it is to the ACE2 receptor. And more importantly, this had a charge switch on the amino acid. So the amino acid went from, I believe it was from a positive to a negative charge. And I just wanted to think for a minute, if I took 2 magnets and I held their north ends together, they would repel, right? But if I switched one magnet, switched the charge on one magnet, it would attract. Right. So I don't know what is going on here, but we know a charge switch is very important in terms of amino acid interactions, and that may have been part of it. We also knew that the EEK mutation was responsible for many of the monoclonal antibody therapies to become no longer effective. Now, a vaccine-induced immune response is a polyclonal response. You get antibodies. Remember that the apple I'm dipping in the chocolate, you get antibodies all over the place. So a single mutation is unlikely to throw off vaccine-induced immunity because it's a polyclonal response. But a monoclonal anti-therapeutic response is different because that's a very specified tool, and a single mutation could indeed cause reduced effectiveness of the monoclonal therapy, which I think was kind of interesting. Also, a number of mutations in the N-terminal as well, but the 351 was a little bit spooky. Shortly thereafter came the P1, which was identified in Brazil. Very similar, had also a collection of mutations, the RBD, some new ones. And these got a lot of people's concern, including ours. And so we started to say, well, why don't we use this mRNA technology where we can change the composition of the virus with a computer sequence, just hit return, change the sequence and then hit return, and then you can produce a different virus using the spike protein of these variants now. Maybe this is a way to ensure that the vaccine is always relevant against what's circulating. So way back in January, actually, we put into the clinic vaccines based on these variant sequences of the spike protein. And we called one of them the mRNA 1273.351. So obviously it's the South African variant, right? And put this into the clinic in an early study to evaluate safety and immunogenicity, of course. And then we did a more definitive study later where we actually had a comparative study where we not only developed this vaccine based on the South African sequence of 351, but we developed another one called 211. And the 2 is important because it's actually a combination. It's actually the Wuhan strain plus the South African, a multivalent vaccine. And we think this may be very attractive because you're not only stimulating the original immunity induced by the Wuhan strain from the vaccine that all of us got, but you're also giving the immune system a sneak peek or preview of what's to come. In this case, it was the South African strain. Now, the 351. Now, the virus is continuing to move ahead of us, so there will be an opportunity, I think, to continue to modify the vaccine as needed to keep pace with the variants. And that's a whole science into itself, and nobody really knows the complete answer on that yet as to how far we have to go, how much we have to go, and so on. We're very actively investigating it right now out of an abundance of caution to make sure that we have the vaccine that we need when we need it. I don't think anybody wants to be back in that situation we were in early February, January 2020, where we literally were defenseless, or we only had social distancing as a way of dealing with the threat of the SARS-CoV-2 virus. So anyway, we're moving ahead, and we're trying to develop these variant boosters or variant vaccines, and we will follow the science and epidemiology and the recommendations of the public health authorities as to when and if they're used. And here, I think this is, I just want to show you this because this gives you an idea of the effect that the variants had on the neutralization antibody titer. What you're looking at here is the neutralization. So the higher the number is, the better the vaccine is able to neutralize what's coming. And the convalescent serum here is shown here in the purple. And what we were seeing is that with the 117, the alpha variant, the one that had swept through England in particular and then came to the United States with such alacrity and speed, we had some decrease in neutralization, but it wasn't as dramatic as we've seen with others. When we looked at other variants, particularly the South African variant of 351, this is what got us our attention in a hurry, was that, and actually I'm going to show it to you on this slide. I think this slide here shows it the best. Yeah, this is taking our phase 1 sera and challenging these people who had been immunized twice with M1273, the vaccine that's in use. And then this looks at their neutralization titer against the G strain, that's the D614G, the variant that emerged in early 2020. That's sort of the standard. And then this was the neutralization titers against the South African strain of the B.1.351, or better known as the beta variant now. And what was remarkable was this fairly dramatic reduction in the neutralization titer. And that got all of our attention in a big hurry. This lower dotted line here is not the minimum level. That's actually the lower level of the assay quantifications. That's the limit of the assay. And what this told us was we saw decreased neutralization titers, so there was something different about this variant against our vaccine than the standard virus. However, it was still able to neutralize it. So we felt that it probably is still effective clinically against the variant, but is doing so at a lower neutralization titer. And that's important because we know that the titers of any vaccine-induced immunity decrease over time. Even though ours, I showed you that data earlier, stay up for 6 months, but they don't stay up forever. And so we're now thinking to ourselves, well, we're dealing with two sort of problems. One is that this serum here, this is as fresh as it gets. This is somebody who was just immunized, and so at their peak. And if I challenge that serum, that person, with the beta variant, we're already seeing a decrease in neutralization just to start with. But if I factor in time as well, that's going to go down even more. In fact, the data show that the reduction in neutralization against the variants gets worse over time than it does against the original strain. you would expect that the vaccine is probably works its best against when it's strain matched. But when you get a variant, it tends to decrease over time. So this is actually what we're all sort of looking at right now, is that with time, we see two things. One is that we know that antibody titers wane with time. The second thing we know is that the neutralization titers against the variant starts off lower. So we have two things working against us. And this, of course, is the discussion and interest right now in trying to deal with this problem with either a variant strain match vaccine or a booster or whatever. I don't know what it's going to be yet, but we're all watching this closely. At the moment, these titers here would suggest that the vaccine retains its ability to neutralize, so therefore should be protective. However, time will tell. Okay, and that's what I'm saying here, is that the mRNA 1273 retains its neutralization activity against 351 variant, suggesting that we would still see the levels are still above those that would be expected to be protective. Teen Cove, so we did study the vaccine in people aged 12 to less than 18. It was very similar to the adult study. We used 100 micrograms dose, the adult dose in the adolescents. And that study we have had reported the interim data out. Looking at that data, it was actually very encouraging. We saw 0 cases in the people who had been given the vaccine, 4 cases in the placebo group. That gives a point efficacy estimate of 100% using the adult definition for SARS-CoV-2, using a slightly more looser definition. The CDC case definition goes to 93. It was well tolerated. Safety and tolerability profile were very consistent to that of the phase 3 in adults, suggesting that this could be a good option for adolescents. And those data are under review right now by regulators worldwide. I'm pleased to mention that the European regulators have recommended the use of mRNA 1273 in this population based on these data. We're also moving into children. We know that children do get COVID-19. They tend to get a less severe illness, but not all the time, and they can transmit it to others. So with that in mind, we went ahead and did develop the vaccine for use in younger people, actually all the way down to 6 months to less than 12, using a dose de-escalation to use a lower dose in these younger people. This study is underway and being enrolled right now. You know, time will tell for the results. And just in the closing here, I'm going to get ideas where, you know, that one thing that's kind of attractive with the mRNA is its manufacturing ability. For a standard biotech company, you'd have a bioreactor sort of this size to produce the kind of ingredients that we need for the vaccines. Here's a person here carrying a beaker. For the mRNA platform, as you can see, just visually, it's a little smaller. And so smaller means lots of things. One means you get more of them, right? You don't need a big building. You don't need as many people. It's faster and it's simpler. And, you know, every time you take steps out, you increase the, you know, the ability to produce the product on a regular basis at the quality and safety and purity standards that are required. So there's, you know, the clinical manufacturing can be quite small. The commercial manufacturing is a little bit larger, but it's at this level, and that's really important, and has allowed us to scale up so quickly to meet the demand for the vaccine. And we're working around the world. We have this just shows we have suppliers and partners worldwide to help produce the vaccine, do fill and finish, and so on, to deliver it to the people who need it, like us. So with that, I'd be happy to take any questions. I really appreciate your attention. Thank you.
First, I want to say, again, thank you, Randy. And I don't think I said it enough, that you're right. The work that you guys did, your colleagues did, that's the reason we're here to get here. It's really something to kind of be in the middle of this kind of return of civilization. So thank you very much for that talk. But, Bruce, there's one other thing I wanted to say. I forgot to do our little commercial for the biotech that of course, we're nonprofits, so freely, but, and willingly, with Gene looking at me, but we have 2 graduate programs that are starting this fall that are supposed to tell you about. Please just check out the website, the Bloomberg website, about it. There are graduate programs, one with St. Joe's and one with Harrisburg University. One's a business degree and one's in biotech. No question, proof. Yeah.
Yes.
So, and excuse me, this is also on WebGen. And
sure. Thank you.
In your clinical trials, what what was the composition of the placebo?
Sure. It was a saline injection.
So what do you think that say the nanoparticle without the mRNA would be a better placebo?
Potentially, but that was also new. So we had to measure it against a standard. And a standard would be a placebo. It's interesting when we looked at the reactogenicity in the placebo arms. There was actually a fair amount of malaise, headaches, fevers in the placebo arm. And not only that is there was anaphylaxis has been reported with the use of the mRNA vaccines. It's in our label. It's in the label of other vaccines. There was anaphylaxis in the placebo arm.
With just saline.
Yeah.
Yeah. But there's a multi-component nanolipid that's used there, so there could be reactions to it, like in other vaccines where they have egg whites and whatever.
Sure, of course. The thing is, what's different is the lipid nanoparticle is a pure chemical manufacture. It doesn't really have classic proteins in it. The anaphylaxis style reactions that you have seen with other vaccines tend to be related to the protein, though it's never been proven exactly that those proteins are the cause.
That's right. That's one of the questions. So could you talk about the reactogenicity of the spike protein?
Sure.
There must be information on that for different vaccine platforms.
Yeah.
Are people reporting the same adverse risk?
Yeah. I can't compare one clinical study to another. So I can't say they got this level, they got this level. We do know that the mRNA is reactogenic, and it's probably because of the stimulation of the toll-like receptor or innate immune system, which actually is a desired effect and it's how natural infection goes by its business. And from my previous life, when I was working on development of Heplicib-B, the 2-dose vaccine, the adjuvant that we used was actually a direct stimulant of the toll-like receptor, the toll-like innate immune response. So it was a desired effect. And that was using an exogenous, another agent, right? So with the mRNA, you sort of get that with the product. It's like, 2-for-1 shopping, 2-for-1 deal. Right? You get the toll-like stimulation with the same product you're using to actually, make the protein. So you can modulate that a little bit by changing the pharmacological properties of the mRNA. You can modulate the amount of stimulation you get. But what's important here is that we generated sufficiently potent immune response to get the neutralizing antibody titers above the convalescent serum and the IG and the binding antibodies, so that we would be confident that we were generating the kind of immune response that would result in the clinical efficacy study we saw.
I was just wondering if you would get more reactive, like in terms of side effects, because you had all these components. That was the reason why I asked.
Are there other? Let me just go because we have to.
Sorry, Sherry.
We'll talk more at lunch. You're staying.
Okay.
Are there other questions, Pat? Sure.
Hey, Sherry.
Hi. So in the beginning, you talked about how quickly the process went, which was incredible and talks to the power of science and prioritizing science. But also, can you talk about what has been done with this mRNA platform previously to allow you to start something?
Sure.
Just repeat the question briefly.
Yes, sir. Yeah. So the question is I showed how remarkable ability to get this vaccine through the 3 clinical studies, but and I mentioned this, and the question was what went in to get us to that point to allow us to do it so quickly? And it was, Moderna had been at this for 10 and a half years and doing mRNA, making vaccines, making other products using the mRNA technology. So a lot was understood about what needed to go into the mRNA, how you needed to modify the mRNA to make it give the right amount of toll stimulation without too much, and things to make sure that it goes to the cytoplasm, express the protein of interest, and so on. You can alter the properties of mRNA to change the duration of expression and things of this nature. That all had to be worked out over time, of course, with careful experimentation. Then we had to go on to then develop the lipid nanoparticle delivery vehicle, because lipid nanoparticles had been in development for some time, but there were problems with them. They didn't do what you wanted them to do. There were stability issues. There were all these, this is all in the literature, dating back for some time now. That all had to be worked out. So that came together. And then we were able to put these two together and put them in and make, I think we made 9 vaccines. One was a combo vaccine. So you could argue we made 10, whatever, 9 or 10 investigational vaccines were developed. So anytime you start doing that, you learn a lot of information about How much dose do you use? What does the structure of the mRNA need to look like? What does the LNP need to look like? Stability profile. How long can it be stable? What temperatures do you store it at? I mean, ours has a very favorable temperature storage profile. That's not an accident. That was worked out over 10 years to get there. We didn't just get lucky. That was hard work to get to the point of having the conventional refrigerator being able to store this product. And of course, we're continuing these efforts to make it even more friendly.
Yeah, so it's a follow-up to Sherry. So this 1 year timeline.
Sure.
A miracle, right?
Yeah.
So how's that going to influence your future product
Sure.
development where you're not getting the de-risking from the
Sure,
government?
sure.
Can you just quickly repeat?
The question was, the observation is that our the overlap in the clinical studies was possible partly because it was de-risked, financially de-risked by the government. For future products, that will be a challenge because I don't expect that we'll see that, and
You have confidence in the technology.
well, absolutely, and that's why we have 23 or 20, depending on when you count them, at least 23 products in development right now. And we've also learned a lot with COVID-19 vaccine development, all we had to go through, and the regulators have learned. So that's, we have to educate the community as well. So I think the world's just in a very different place. But yes, every new product will go through the same safety and efficacy rigorous analysis. We absolutely expect that, and we want it, by the way. We want that kind of a rigor.
So,
Yeah, John.
question. What, if any, data can you share about the vaccine, the mRNA vaccine against the Delta variant? That's all we're hearing about.
Sure. So what I can say, we have published some data in a bioarchive preprint edition where we did do some neutralization studies looking at how well does mRNA 1273 neutralize Delta variant, the B.1.617.2 or Delta variant. And it's very interesting. The reduction in neutralization with the Delta is fairly modest. It's on the order of 2 or 3-fold reduction. What we saw with the South African variant or the B.1.351 of Beta variant was more like 7 or 8-fold reduction. So it's less, the neutralization is less. The reduction in neutralizing antibody titer is less with the Delta. I don't know I can't translate that into a clinical story and to say that this does that. It's just that we would expect that we would have efficacy at that level. Yeah.
Tom,
Yeah.
Randy, could you comment on what's needed to move from emergency authorization to full authorization?
Sure.
for the FDA at
Yeah.
this point?
Yeah. Well, I think the primary is length of follow-up. That's really what it was, because the vaccines were made available for emergency use with 2 months of follow-up. Okay. At the time, that's what we had. Of course, we had a lot of follow-up on previous mRNA vaccines developed for other diseases I've already mentioned, but that of course, wasn't COVID-19 vaccine. So I think it's just duration of follow-up is one of the primary criteria that the agency is using. Of course, I don't work for the agency directly, so it's really finally up to them. But I do believe length of follow-up is one of the key criteria.
Thank you. The last question, I want to ask this from the web. Sorry, Bruce. We'll talk more at lunch. You said that the government, U.S. government, guaranteed purchase of the vaccine if you met minimum criteria.
Yeah.
What was the minimum criteria?
Well, certainly one of the most principal success criteria was efficacy, and that was that the lower bound would be 30% or greater.
30%. Aim high, U.S.
Yeah.
Sorry.
Yeah.
But 30%. Think of the difference. Think of the different world we'd be in if the vaccine was only 30% efficacious. Wow.
It would be a very different world. No,
I think with that, I'll thank Randy again for a great talk, really historic talk, and thanks for making it here. And thank you all for listening and participating. Those things for lunch, we have the lunch hidden behind those rubber, so we'll unsheath them right at the back of the room. Obviously, washroom across the hall. So if you're staying for lunch, please, I think you can take your mask off when you eat lunch. Get your food, come back. And Randy's meeting with the students at about 1:15. I think that's it. I wanted to thank Tom and the production crew and Ed for making this a big successful event, and everyone else is putting on WebJam. I hope it's being captured. We had about 500 to 600 people online.
Cool.
Thank you very much.
Yeah.
Thank you.
Great book.
Thank you.